Amy Murray developed a zebrafish aquarium in her classroom and enabled 16 of her AP Biology students to compete in the Pittsburgh Regional Science and Engineering Fair, doing some fairly sophisticated experimentation on zebrafish embryos. For her fellowship, Mrs. Murray and her AP Biology class were able to spend a day on a STEM career tour. They began the day at Novium. Having just learned about cell interaction, they were fascinated by Novium's research and product development involving paracrine signaling, the process by which human cells communicate with other nearby cells in order to keep them healthy and functioning properly. The students were given a challenge: use the multi-channel pipette to quickly transfer fluids. What appeared to be easy for a seasoned professional proved to be more cumbersome at first for those with less experience. This wasn't the students' first hands-on challenge of the day. Outside the clean room, they were instructed on the proper way of donning biosuits and the delicate task of avoiding contamination. No touching with bare hands, no contact with the floor. I already failed, laughed Alex, the senior, as he and his fellow students managed to suit up and pose for a round of selfies. Throughout it all, students learned how science is used in actually helping people. The tour included a stop at Sharp Edge Labs, a small company who specializes in discovering drugs to treat genetic disorders of protein trafficking. Sharp Edge Labs has launched a patient-driven program with the purpose discovering drugs that prevent or reduce the effects of faulty protein trafficking due to monogenic diseases. Mrs. Murray walked away with a statement: "This actually changes how and even what I teach." Sharp Edge inspired all the students as they explained how they're working on new treatments for diseases such as cystic fibrosis, muscular dystrophy, and Gaucher's disease. Stops on the STEM tour also included Ishima Structural Biology Lab with nuclear magnetic resonance spectroscopy. Dr. Ishima oversees a team of research associates and fellows who are currently working to determine protein structure and dynamics using nuclear magnetic resonance. We were all amazed at the massive size of the magnets in the lab used to visualize proteins that are smaller than human cells, than even the nuclei of human cells. We were also led into the X-ray crystallography lab. Here, researchers are able to grow, store, and monitor crystals. Once the crystals are ready for analysis, X-ray beams and image plate detectors are used to collect data about protein structures at the atomic level. The high point of the STEM tour came with a visit to the Sang Lab in the zebrafish research facility. A brief presentation made us aware of the scientific breakthroughs these amazing fish make possible. We were escorted into a room of thousands of aquarium, each labeled with the experiments running, and various graduate assistants updating data logs. The eggs of zebrafish embryo were transparent, so you can study their development. Inspired, Mrs. Murray created her own zebrafish aquarium and challenged each of her AP Biology students to develop experiments worthy of the Pittsburgh Science and Engineering Fair. Hard work ensued, even after school and lunches, until the big day came for the teams to go to the PRSEF. Topics included the effects of supplements on fetal alcohol syndrome, the effects of lithium chloride on the embryonic development of zebrafish, the effects of acid mine drainage, the effects of estrogen on zebrafish reproduction, and the effects of nicotine on zebrafish development, the effect of temperature on zebrafish embryos. Congratulations, 2017 Grow a Generation Distinguished Educator and North Catholic Biology teacher, Amy Murray. Great work.